Alright, so in this video we are talking about the next function of blood, which is coagulation. And to appreciate coagulation, we are going to talk about it as a property of plasma, and specifically we're going to talk about platelets. So coagulation is simply that it's a property of plasma, and it's a process by which soluble fibrinogen is converted into insoluble fibrin. Okay, so coagulation. So this is a process in which soluble fibrinogen is converted into insoluble fibrin. And that's what we're really going to get at. So terms that we will need to familiarize ourselves with as we go through this component is we need to know what coagulation, of course, is. Not only that, but we need to appreciate what adhesion and aggregation are. And in between this, we will appreciate activation. We also need to understand what a primary and secondary hemostatic flag is. So we have the primary hemostatic flag and the secondary hemostatic flag. And of course we need to understand what a clot is. So that's 1, coagulation; 2, adhesion, aggregation, activation, primary and secondary hemostatic flag, and what a clot is. There are several factors that we will be looking at as we go on. For example, fibrinogen itself is known as factor 1, and we'll go down all the way up to about factor 12. And these are listed for you in the notes that have been put up for you on Moodle. So 1 is fibrinogen, 2 is prothrombin. We'll allude a lot to factor 4, which is calcium. But factor 3 as well, which is tissue factor, will also come into play. There are other factors in between that we will be alluding to, as well as factor 12, which is the fibrin stabilizing factor. And as we go on, we'll be mentioning them. Aside from these 1 through 12, actually 1 through 13, 13 being the fibrin stabilizing factor, I beg your pardon, we will be looking at pre-kallikrein, which is also known as the Fletcher factor, as well as the Fitzgerald factor, contact activation factors, and we will just appreciate how all of these come into play. So when you look at a platelet, this fragment has several receptors that include collagen receptors. So we have collagen. We also have ADP receptor. And then we have von Willebrand factor receptor, and then we have fibrinogen receptors. Within the cytoplasm, this is among others. Within the cytoplasm, apart from lysosomes, we have dense granules. We also have ADP and calcium, and we have several other factors. So we have dense granules, we have alpha, so these are the dense ones, Then there's the alpha granules. We also have on the surface, apart from ADP, collagen, von Willebrand, fibrinogen, we also have leukocyte receptors. So in particular, P-selectin. The other things that we have here, for example, the collagen receptors are known as GPIB. All right. So let's just go through some of the mechanisms and use some of these factors. I would want to emphasize right now that aside the dense granules with non-protein substances like serotonin, we also have another type of granules, which is the alpha containing granules. So we have surface, we have cytoplasmic components, we also have secreted proteins. So secreted proteins will be referring to things such as platelet-derived growth factors, and some clotting factors such as 5 and 8 and fibrinogen, so on and so forth. So to better understand the science, we will look at the endothelium itself. So these are the cells that line the vessels. We'll look at what does a normal endothelium look like, and what does it look like when there is injury, when there's when there is a penetration, so to speak. So the first thing we need to know is the circulation of blood is in liquid form, and if there is any injury to the vessels, it forms a plug, all right, so that the circulation is not interrupted. And this mechanism for prevention of blood loss is the one that is termed as hemostasis. Now, in a healthy circulation, we have several mechanisms that ensure that blood is kept in liquid form. First of all, we have the fact that it is anti-platelet, and then we also have some mechanisms that are anti-coagulants. Then the next one is where we now have the fibrin inactivation mechanisms. All right, we don't want platelet coagulation or fibrin formation. So these are the things that we're going to appreciate. So first of all, we want to see the anti-platelets. So in anti-platelets, we are really talking about the endothelium acting as a physical barrier between platelets and the extracellular matrix that is found under the vessels. All right, so these are the extracellular matrix. First of all, what we have is the endothelial cells themselves acting as a barrier between platelets and this matrix. Okay, the endothelial cells in the capillaries also produce chemicals. So first of all, we have the barrier, and then we see that there is a chemical production. Okay, so this chemical production is to maintain the liquidity or the fluidity. So we have nitric oxide, then ADP or ADPase, as well as PGI2 or prostacyclin. Now, what does the nitric oxide and the PGI do? So these, the nitric oxide and the PGI, do not allow the platelets to stick to the endothelium. All right, and they work by binding to the receptors of the platelets. So to be specific, NO and PGI2, these, what do? They stick to the receptors of platelets. And what does this do? This inactivates. It inactivates the ability for them to bind. So inactivate binding where to the endothelium. All right. Aside this, they also function as vasodilators, allowing blood to flow, specifically nitric oxide. The ADP. So ADP favors attachment of the platelets to the endothelium. So where there's ADP, you find that platelets will bind to the endothelium. So when you have ADPase, what it will do is it will break down ADP. Therefore, if ADP is broken down, it means that we won't have this binding taking place. Therefore, in a healthy endothelium, there is really no allowance of platelets binding to the endothelium or even to the matrix under the endothelium. The next one that we're going to look at is anti-coagulation. So in anticoagulation, we see that healthy endothelium keeps the coagulation factors at a low concentration. So let's start with heparin sulfate. So heparin sulfate is expressed on all endothelial cells. So what does it do? So we have heparin sulfate. What does it do? It binds to antithrombin 3. Okay, so antithrombin 3 comes from the liver and is present in blood. So it will bind to antithrombin 3. And when it binds, what do you get? When it binds antithrombin 3, it is activated. And in this state, it then can inactivate clotting factors. So once there is this bond, then you will see that there is an activation that will take place. And this activation is then going to inactivate clotting factors. And these clotting factors are thrombin and factor 10 as well as factor 9. We also have another one. So this was the first one. The second one that we have is known as Thrombo Modulin. So Thrombo Modulin is also expressed on the endothelium. So what does it do? Thrombo Modulin binds to circulating thrombin. So once this binding takes place, you see that it modulates the thrombin. How? By allowing it to activate So it activates protein C. All right, now protein C has the ability to inactivate factors 5 and 8. So it's a cascade. So in the first one, heparin sulfate will bind to antithrombin 3. And when antithrombin 3 is activated, it will inactivate thrombin as well as factors 10 and 9. The Thrombo Modulin also modulates thrombin by when it binds to thrombin and allows it to activate protein C, which then can inactivate factors 5 and 8. All right. The next one that we're going to talk about is fibrin inactivation. That's number 3. So in fibrin inactivation, we see that if fibrin is in circulation because of anything, maybe there's a clot at a point, and then it circulates to a healthy area. We see that the healthy endothelium can release what is known as tissue plasminogen activator. So TPA, what it does is it activates plasminogen, and this comes from the liver, all right, and it converts this to plasmin. Now, what does plasmin do? What plasmin does is it degrades the fibrin. But not only does it degrade the fibrin, but also other factors such as factor 5, factor 8, factor 12, fibrinogen as well, and prothrombin. Okay. We also see that protein C here may also activate TPA. So that is something that we can see. So we have protein C also having an influence on activation of TPA. Alright, so this is what is happening in a healthy endothelium. In the next video, we'll try to appreciate what is happening in an endothelium that is not healthy, or that has been infected, or something has happened. And let's see how this clotting or coagulation goes on in order to keep blood fluid.